benvinguts, bienvenidos, bienveni, welcome. It's a real pleasure to have you here. Just let me recover some notes. My name is Josep Lluís Pelegrí. I'm the director of the Institut de Ciències del Mar and the Centre Mediterrani Investigacions Marines, which holds not only the institute but also the unit of marine technology. And in this building we also have the Institute of Evolutive Biology. So in behalf of the three institutions, I'm extremely happy to give you a warm welcome. I'd like to first welcome our speakers, Colombón de Vargas and Andreu Abreu. It's really nice having you here. I'd like to thank and welcome Silvia González Alcinas and her team, all the past and present researchers that have been extremely involved in Tara Oceans from the very beginning. They are one of the 7 pillars of this project, as far as I know, so thank you for this participation. This has enhanced and has projected our center worldwide, so extremely happy about this. We are all thankful to your work. I'd also like to thank the Gilles Mirambeau and our colleagues at the PRBB that they have participated in the organization of this event. Also people from the Club Pativela, you see the Patinaires, the Catalan sailing boat that it's upstairs also, and thank you, they have also helped a lot. And a warm welcome to the Tara crew. And especially a very warm welcome to all volunteers. This one of the assets of this project has been to mobilize people, and because of its characteristics. I'll say a few words about that later. No? But we are extremely happy and thankful that you are here and helping. This wouldn't have been possible without you. No? And so we are very happy to be part of this celebration. Thank you for bringing us in. This mission, in our view, has several exemplary features. No? One is that it has created synergies among many research centers. This has been extraordinary. No? Many centers have been involved and are involved in these Tara projects, and this is a great example for all of us. So this is something we have to follow. Another result has been obviously the great amount of science that it has produced, from genomics to taxonomics, and it keeps going, no? And they are opening many new facets, so this is excellent. No? And particularly also the third asset, I think it's worth mentioning, it's been an extraordinary example of sustainable development by doing research in a sailing boat. A sailing vessel. This is particularly important, I think, nowadays. No? And this brings me to just a very brief reflection that I think is important to make loud and clear. No? So we are facing very critical times. This is a time of global change. It's been for several decades already, and it's a time of climate change. We know that the weather is changing, that average temperature of the Earth is already increased 1.2 degrees since the pre-industrial times, and we are faced to keep it below 1.5 degrees, hopefully, but there's very little margin. If we just keep producing or consuming fuel oil or fuel gases, and at today's rate, we only have 10 years left, and we'll have exceeded the amount of carbon dioxide that has to be in the atmosphere to keep below this 1.5 degrees. So everything is showing that rather than keeping it or decreasing, it's going up. So in a few years we'll have no margin. No? So and not even the 2 degrees. The prospects are that we are going to go far beyond that. So this situation, we wonder, and I wonder personally, and we wonder as a research institution what we can do about that. No? First of all, I think we have to keep our role of scientists, study and identify what is the current situation of the living Earth, what are the threats to biodiversity and humanity. This is possibly our task, no? As research scientists, and it's probably what we expect from ourselves, but this is not enough. We have to change our view of what is our role in this situation, and we have to do it together. We have to take a position. So we have to communicate the situation and the dangers and do it loud and clear with no fear. That's the first role that we scientists, sometimes we try to be accepting, no, we try to take no positions, we try to say things only when we are 100% sure, but this is not possible any longer in this today's situation. No? Second thing, I think we have to teach through example. Using a sailing boat probably is one of the best examples of doing sustainable research. But this goes to everyday life for each of us, no? in our everyday life, and to reduce, to first of all, to reutilize and to recycle. This is the 3 R's, no? And we have to go this way and teach everyone to do this, and teach society, teach our kids. Our kids are mobilizing us, they are moving, but sometimes I fear that they don't have not yet time to interiorize this situation, interiorize what they have to do to face this danger. No? So I think this is the first thing. We have to, together with them, teach them that we have to reduce. There are some things we cannot do any longer. We cannot change our mobile every year. We cannot travel to Paris by plane. We cannot consume every single series that Amazon's or whatever, or Netflix produces. This is not. And third thing is we have to join forces with society. Tell them and listen to them, no? Create associations, neighborhoods, protection of the environment. I think that all volunteers here are probably the best example of the power of associations, no? Without volunteers, without this fast response and their will to get together and organize, this wouldn't have been possible. It's been you, no, that made it possible in just a few days, and how Tara is such a claim, no, for you to react, no, and ICM we want to be a change, or a claim also. But we have to do it like by through everyday action. So we have to join forces, no. And finally, the last thing I think we have to do as a position is we have to realize and we have to convince people that it all comes to justice. This is a situation of global justice, social justice. We are developing ourselves, and there are many people, there are many countries that are suffering. Every time we purchase something, a first world product, there's people that in the third world countries are suffering, are being exploited, and their environment is changing, and they are the ones that will not be able to face all these changing environments. We probably will be able, the USA will be able, and all these producers of the carbon dioxide will be able, but they will not. And the difference, the gap will keep increasing. So if you love this earth, you have to work for social justice. I think this is the final message. So I close here. Again, thanks to everyone for being here, particularly the Tara people. Thanks so much for your example, for your synergies. And thanks to all the volunteers. We are extremely happy to celebrate this 10th anniversary with you. Thank you.
Yes, thanks to these very inspiring words. It's hard to go back to concrete aspects after this global overview of the challenges we are living. Thank you very, very much. So hello all, I'm very pleased to be here. I'm André Abreu. I'm the head for international policy for the Tara Ocean Foundation, working with Tara since 10 years now, like the Tara Oceans project a bit. I'll try to present briefly the foundation, the boat, and how the foundation became this community to host and to welcome scientists from the entire world to develop innovation and ambitious scientific programs that sometimes are hard to do with just one institute. So this synergy, this multidisciplinarity, Mr. You were mentioning, is it's a bit the seed of the Tara program. And after Colomban prepared some some videos from 10 years ago where he's very young. Sylvia, she's pretty young also, and it's kind of, comment on dit, well. So Tara is a boat that was built by a famous French explorer, a very extraordinary guy called Jean-Louis Etienne, which was the first man that crossed Greenland alone by foot without assistance, the first man to be close to the North Pole without assistance also. So he built Tara 30 years ago, exactly, 30 years ago in 89, with the dream to repeat this incredible history of Fridtjof Nansen and the Fram. You may know that in 1887, this crazy Norwegian young researcher, sorry for my English, had the idea that the North Pole was not a continent but just a frozen ocean, and this was against the common understanding at that time. He was quite attacked by the scientists at his time by this proposal, so he decided to build a boat, to put the boat in the ice cap in the North Pole, and to see if it drifts or not. So she had the supposition, the idea that there was a transpolar current crossing the Arctic from north to south, from east to west, and so he did the Nansen. And so he did the drift, and he was the first man to be in the North Pole at this level. And 100 years after, Jean-Louis Etienne realized that no other sailing boat had repeated the adventure of Nansen, so he decided to build a boat to go to the North Pole. So it explains why Tara is this whale form, this la baleine in French. It's very round. It's to escape from the ice. When the ice closes in the North Pole, it lifts the boat and not crushes the boat. So Jean-Louis Etienne didn't succeed to do the drift for many reasons, but mostly financial reasons, because it's quite a logistic and it's a challenge to put the boat in the ice. He sold the boat for a famous sailorman, Peter Blake, Sir Peter Blake, champion of the sailings, that create a foundation with the boat. He renamed the boat Seamaster. It was named Antarctica before. It was renamed Seamaster, and with a spirit of protect the ocean. So he created a foundation to carry expeditions, but with a will to protect the ocean. So this policy and this awareness and outreach aspects was really the core of the project of Blake. At Blake, there's a tragedy. He was shot dead on board of the Tara boat going up the Amazon River by a pickpocket. You know, it's a sad history. And then the boat was in New York without use, so the widow of Peter Blake asked Etienne Bourgois, the son of Agnès B., if he could be interested in to continue this dream of Peter. And Etienne knew also Jean-Louis Etienne, so he bought the boat 17 years ago. And he created the Tara Foundation to join the two spirits, the adventure spirit of Jean-Louis Etienne and the humanitarian and environmentalist spirit of Peter Blake. So Tara Ocean Foundation is born like to marry and to join the spirit of go beyond the limits with this boat that can go everywhere, and to work to protect the oceans and to make big science. But big science arrived in the Tara project step by step. So I'll tell you some steps. So, but it was just an introduction. I'll go fast with slides.
I'm joking, but I think you are all invited after a while, right? So that's important. After our little chat, we can go to the redfish.
We can at when the people leave the auditorium, they will have a card in their hand, and with that they will have free drink at the redfish.
Free drink at the
We,
With the card
we will give the message after. So this is the spirit of Tara, so big expeditions beyond the limits of the usual funding today. We have to say that a day on the sea for an oceanographic boat, a big one, is very expensive, from $30,000 to $50,000. And so to create an expedition of 3 years in a big oceanographic boat is quite a challenge today. With Tara, the costs are reduced, the footprint, the ecological footprint is reduced, so we can dream, and we can project expeditions for 3 years and more. So these expeditions with high-level research, of course, the education and the outreach aspects are in the project since the beginning. The media partnerships, the partnership with schools, bringing the kids on board was since the beginning a quite strong will from Tara founders, and it's going more and more, and it's really cool. And at the end, cooperation and advocacy to protect the oceans. We are doing what we can to participate on the climate negotiations, to participate on the high seas negotiations, to help to design protected areas, and to try to push the ocean governance to real laws to protect the oceans. So we can talk about 3 major axes and programs of research. The climate one that started with the Tara Arctic, the Arctic Drift, the biodiversity, the plankton and coral reefs issue, but I think plankton is the heart and the spirit of the Tara project still. And now we are also tackling the issue of plastic pollution in the ocean, microplastics and now nanoplastics, because we see more and more plastics when we sample, and it's simply, we cannot just go back to our homes and sit and do nothing. So we decided to act, and we are doing quite a strong advocacy against plastics. I'll say some words at the end. So we tried, Colomban will explain all the scientific aspects after, of course. I'm not a scientist. I'm a policy guy, so won't enter into science, but this concept, global ocean systems, ecology and evolution, it's a beautiful concept that I think brings together all the research Tara is doing. So, the major expeditions, this crazy expedition in the Arctic from 26 to 28, 507 days in the ice. 6 months without any sun, without any light, any natural lights. No assistance possible in this period of time in the Arctic. No helicopter, no nuclear submarine, nothing. If there is a problem, you're just fucked. So have to be. Yeah. So. This is Tara caught in the ice, in the banquise. This is the comparison between the Fram in 1893 with the Tara almost 1 century after, even more. And you can see that we did very close to the North Pole. It's still the boat that went more close to the North Pole ever. I learned this with Chris Ball recently. Still, Tara is the one that went more close to the pole. But the information which is terrible here is that the Fram took 3 years to do this, and Tara took 1 year and a half. It's because the banquise, the ice cap, is melting, so it goes more and more fast. Actually, the banquise, the ice cap, is running on 10 kilometers per day. So this is the Fram. This is the Tara in the night, in the polar night. Thanks to all the photographers and artists on board that follow us with real incredible and amazing stuff, and you can see it continues and it don't stop with the Plankton Planet project and beyond. So this was when we changed the team in the boat. So there was a plane that brought the new team. And after still, the Tara was still in the North Pole when Eric Arsanti, a very, very small guy, but with so bright eyes that you cannot imagine, he phoned Etienne Bourgois, our president, to propose to have an expedition around the world to follow the Darwin Beagle story. And then Tara Ocean was born with a group of scientists. You see the vintage images from Colomban tout à l'heure. And they did 3 years and a half around the planet, around all oceans on the planet, only on plankton, doing the same protocol around for the whole ocean. And actually is the most complete survey on plankton ever, is the most complete database on marine biodiversity ever collected still. So Tara Ocean, I will leave it to Colomban. I'll finish with the more recent expeditions. So Tara Pacific, this was after the COP21 in Paris, where we acted quite strongly for the climate. And you have this El Nino phenomena more and more frequent, so which are threatening the corals, the coral reefs, that are blanchiment, whitening of the corals, the dead of corals. They become white and they lose the algae. So we decided to go to the Pacific, where all these corals are, and to have a complete survey on the Pacific for 2 years on all the reefs we could sample. And we sampled the reef, the ecosystem around, and the ocean around, to study also the plankton and the microbiome associated to the reef. It was, like, I think, the first time at this level. Voila, Tara Pacific. 2 years doing west to west, north to south. We had some problems with some countries that didn't deliver the authorizations, the permits. You can imagine which ones, those who know the research. But this, just a word, because this is a huge job the scientists sometimes cannot do, and Tara Foundation do this job to ask for the authorizations to follow the Nagoya Protocol, due diligence reports, blah, blah, blah. It's quite heavy, actually. It's a full-time job in our foundation. And then each sample we had plastics around. Even in the Arctic, we found microplastics. In the Antarctic, we found microplastics in 26. So we decided to have a plastic expedition in the Mediterranean, but also focused on the interactions with plankton. I mean, not macroplastics, which are quite well understood today, but the tiny ones, the microplastics, and how they interact with the ocean microbiome. This is just an example of how the results are presented. We can have also modelization of these results on the regional sea. We can say that we have more polyethylene or more polypropylene, which can be a source of information to the change. Which kind of plastics we have to ban? We don't know. Which kinds of plastics are really harming the biodiversity, and how? We still don't know. We have these horrible images of the seventh continent, the so-called seventh continent. I have a slide of the seventh continent. You'll be surprised. So this is a kind of modelization that can be a fruit of the results of the data. Just to say that there's also, we found important another threat, that these tiny plastics can be a transport for species, even for viruses, bringing viruses from the south to north and etc. And so it's also also an issue. So this is the the plastic continent. Okay, we were in the heart of the so-called Great Plastic Garbage Patch in the North Pacific, which some people think it's a continent of plastics, but actually no. It's just a soup of tiny, tiny, tiny particles of plastics. You can see them. The Mediterranean Sea has almost the same concentration, actually. So there is the message we try to deliver is there's plastic everywhere, and it's not one magic project that will clean the oceans and that we can put stuff on the middle of the garbage patch and then to collect the plastics. No, it's really not feasible. Plastics are everywhere, and the vast majority are in form of micro and nanoplastics now. So it's feasible to clean the ocean. If you clean the ocean, we clean all the ecosystem with, actually. There's a French writer that is writing about the Anthropocene, that is talking also about plastisphere, that we created the new ecosystem where plastic is a part of it, and we cannot change it anymore. So we have to study to understand how it's interacting with biodiversity. Just to end, some message on the advocacy and on the big challenges we have today. So we have some attachment to the open data. Since the beginning, all the data collected from Tara is open source. The project is the projects are built on the basis of a bottom-up cooperation among scientists. So they decide how to design the expeditions, what are the main questions we are willing to respond, and all this data is open, and there is also a will to collaborate with other countries, to include more developing countries in our studies. This is an issue because there's a lot of capacity building that is needed to invite countries from Africa, for instance, to participate on this kind of programs. So free access and the sharing of data and all this issue about the transmission of this. I'll just, before giving the floor to Colomba, just a final message on the protection of the ocean we need, the climate crisis, and just to say that 2020, we are now entering in a very strong momentum for the ocean on the political side. You know that the COP21, the Paris Treaty was signed, but still not implemented, and the ocean measures are still to be included. We are working on it, and in Chile, in December, in Santiago, you have the COP25, where the Chilean government is calling for a blue COP, a COP that will address the ocean issue, definitely. We just had the release of the special report on ocean and cryosphere by the IPCC. That was a strong text, strong messages. We are bringing this to Santiago, and we are trying to make a lot of noise in Santiago that these messages from IPCC will not finish only on our USB keys, but will be followed by concrete action, funding for science, funding for research on the climate side, because yes, we need to understand. We talk about blue carbon, but we don't know how to assess really the carbon pump in the ocean. There's a lot of gaps that must be filled, scientifically speaking, on the climate and ocean nexus. And so this is one of the big momentum. And from the COP25 in Chile and COP26 in Glasgow in 2020, states are invited to increase the ambition of the commitments, the contributions, and we are trying to push states to put ocean-related NDCs. I'm too technical here, but ocean-related determined contributions for each country. Once they are written, they have to do it. The Paris Treaty is a mandatory agreement, so they have to respect it. And next year, so we have also the UN Ocean Conference in Lisbon. that we push the SDGs, the Objetivos de Desarrollo Sostenible. And then we have the Marseille Congress of the IUCN for the World Congress of Nature. And then finally, in October 2020, we have the COP15 of the Biodiversity Convention of the UN, which will set new targets for the period 2020-2030. New targets for protection of the ocean and concrete measures for creating MPAs, marine protected areas. But all this process needs the best science to be real, to be based on real science. And it's not often the case. I'm sad to say this, but it's often the political measures are not connected with the scientific community properly, or they can do better. We understand this. They can do better. I spoke too much. Thank you all. Thank you, ICM, for hosting this.
Okay, I have largely too many slides, so I will try to check if you are interested in my slide and, you know, and keep only the one where I feel a bit some good energy. So I think I'm not going to give too many. I was not sure to whom I was going to speak. I thought it was maybe more of a grand public, you know, talk, but I recognize a lot of people, scientists. So anyway, so I will try to, maybe I will try to give a few concepts rather than scientific results. To try to understand why Tara Ocean was Tara Ocean and was so successful. Sometimes I think it's a bit puzzling and one of the answers is definitely that. How does it work? Sorry. It was really based on it started with a very small group of people and really around Eric and Steffi. Eric and Steffi was really the node, the core stuff that, you know, that really made this expedition a success, and they were working together extremely well. And Eric was at the perfect time in his career to bring this project. So I think very quickly the dream was to go to the full ecosystem. And I will explain you why as well. So also it was a lot about, you know, it grew totally organically, a bit like, you know, like a biological object. It was just friendship, so it was a lot of people coming just because we needed to be a bit crazy to go for these 4 years, you know, expeditions. So I think it attracted a bit people that were a bit different, kind of brave and a bit crazy as well. So I will, yes, and in fact we are here also 10 years after Tara Oceans, at the beginning of Tara Oceans. So I will show you a few images of this, of the beginning of this expedition. That was me when I was young. So I think we can start here. So the sound is quite bad, but
I would remove the microscopy from this.
So we have 1 year to do the expedition.
Sample.
So in the beginning, we did a lot of these meetings. We are meeting 1 year around Eric, trying to build up this.
Yeah, decide what is the base data set.
Okay. Oh, there we go. Good. Colomban?
You have to make one at the end of the trip.
Just before the departure. So there was a lot of passion and friendship.
Ce qui m'intéresse, c'est quand on a vu la ville, tout à l'heure, dans un litre de mer, il y a entre 10 et 100 milliards de virus, il y a entre 1 et 10 milliards de bactéries.
Pour le temps, il est très beau. Il y a énormément de bateaux qui font la sortie avec nous.
Tens of small boats, you know, following Tara. And So this was picture taken by Gabi. This is the first, we were totally drunk because we had a big party the night before. That's why the camera is moving a bit. And in fact, what was crazy is that the protocol was not ready at all. So we left, on the boat. We had no funding, really. And we had just, put this protocol. The protocol, I think, was quite brilliant, in fact. But it would build it up, while we were at sea. In fact, it was ready when we were in Sharm el-Sheikh, in Egypt. And you will see something very... so Barcelona, October 2019. Huh? This is 10 years ago. It was October as well. I don't remember when exactly. You can see Eric arriving on the Ramblas. And so Christian Sardet took literally hundreds of hours of the whole 10 years. Because in each meeting he was here filming us. So we have 10 years, of rush. And I think if you know someone who wants to do something with this, and this is here 10 years ago. Anyway, a bit of emotion. And so everything really started from Eric, who is an expert in analyzing how life self-organizes, how life takes shapes by collective behavior between molecules inside the cell. And Eric taught us that function emerges from interactions between self-organized shapes. So the shape self-organize by mathematical laws, and then they take shape, and sometimes this shape take a function. And this is really Eric, big message. It's 40 years of Eric's works at the cell level. And then when I met Eric, he showed us this amazing movie that you can see. Actually, this is all cells of a zebrafish dividing. And so you see a baby fish by spin microscopy. And you can see all the cells interacting and taking a shape as well. And so what is crazy, in fact, is that you can mathematize, you can formalize this shaping of life with quite simple equations, reaction diffusions, so mathematically, you can explain very complex things in the shaping of life by relatively simple equations. And this science doesn't need DNA, You don't need DNA to explain this. DNA is just the memory of this. And so Eric has a very different view of evolution than I think most people. And so the dream, and we still are running for this, is why not, why aren't we living really on a self-organized planet? These ecosystems also have a shape, and probably because you can find, equations that explain this pretty well, you can this as well. And why not at another level of organization, where in a sense, organisms would be like molecules or ear cells, right? And so I think we are still running toward this goal. And the beauty of plankton is that plankton is super active, right? It's taking shape and again and again. And so I think it's the best model, for ecosystemics, for global ecosystemics. Much more than all these ecosystems that are super stable, that lived much longer than us. They have very long lifetime. And so it's very hard to study the self-organization of these ecosystems. So we decided to go toward these beautiful systems, which, as you know, is super important also for oxygen production. It sustains marine life, and there is the carbon pump that is super important. So I think the whole thing we did really well, thanks to Eric and to these magics of the people who were there at this time, is to build a protocol that was really holistic and pan-ecosystemic. So we really put together this protocol that target from viruses to animals. And this was really well thought in 1 year. So we had different experts, Matt Sullivan, Sylvia, myself, Gabi Gorski, and then we had people and then we sequenced a lot and then we analyzed different level of organization of the ecosystem, of the system, from genes to express genes to organismal images. So we tried to maximize the layer of the system we were analyzing through the entire life kingdoms. And so I think the protocol was, and we put everything on a little boat, so that was also something extraordinary, thanks to Eric as well, actually that was more to us. We maximized, this protocol on a very small sailing boat. And then we took this exact same measure for 3 years, 4 years in fact, 3 years for Tara Oceans, and then another year for Tara Polar Circle, Tara Ocean Polar Circle. And now we'll have a lot of data from Tara Pacific. Tara Pacific, there will be 10 times more data than Tara Oceans. So just for Tara Oceans, we have 90 terabases of DNA. This is this number from 600 states of the ecosystem. Because we have about 200 sites and 3 depths. So we can say that we measure this huge body that is the ocean at 600 states, 600 times, and with this amount of information, and also quite a lot of imaging. In fact, we have now more than 10 million images. This is the best data set we have on any biome today on Earth. This is the best data set we have. We don't have this equivalent for desert, for forest, it's impossible for us to have the full ecosystem in a forest. And so I think this is probably the best material we have to understand the pan-ecosystemics of a global biome. That's why I think we had this success. And that's why we didn't publish much, and now we are, I think, at 130 consortium paper. In 10 years, it's not a lot when you think about it. But it took a long time, and we were not we were active, but, but I guess we took our time. We were criticized sometimes to not put the data on, on public database, so the Americans were pushing us to put things on publicly. But we took our time, and we produced really good quality data. And the first papers, started basically after we came back, so toward 2014-15. And then I think we did a very, very good publication in terms of quality with this consortium paper. We'll have, this year, we have 3 papers in Cell, which is really a big achievement. I think it's even better than the Science. We did a special issue of Science, but this is really, really important because Cell is really a journal of people studying, cancer and cell biology and medicine. And the fact that Cell is taking 3 big papers in ecology means a lot, I think, to the time we are living. Basically, I think they really want to turn toward environmental science, and so this is really, really cool. So there will be a big noise at the end of the year. The papers are still in preparation. So just a few results, but I should really go fast because So I think, first thing. So don't look at the at the slide. Just listen to me. I think the first thing we discovered, of course, is that there is many more genes, many more species, many more whatever. And we published a lot of papers about, okay, this is much more complex than we thought. But what is amazing is that it's measurable. We are often reaching the limit, the saturation of the number of genes, or the number of ribosomes, etc. So in fact, with the method we have, mainly the high-throughput sequencing, we can see the limit of this complex oxygenic ecosystem. The group that seems to be the more complex are the unicellular eukaryotes. So the viruses seems to be quite simple. The bacteria are a bit more complex. And then the unicellular eukaryotes, there are tons of genes. We are now reaching more than 200 million genes. Most of them are totally unknown, not even comparable to anything that exists in databases. And so there is a big problem, to include in the ecological game the eukaryotes. I think that's going to be a big, big challenge. Just to show you, for instance, we built a tree of plankton life, which is this tree it was built with a little marker called the ribosome, a V9 sequence of the ribosome, and this tree is super solid, in fact. It holds very true, and the group that is the most diverse and very abundant are the dinoflagellates. And this is super embarrassing because dinoflagellates, this is a curve of genome size. You cannot see really, but I think humans are about here, and the dinoflagellates are here. So they have genomes, 110 or 100 times bigger than the human genome, and they are super complex cells. Just to give you an example of this complexity, here you have a little dinoflagellate that transforms its chloroplast into an eye. So it has an eye. It's one cell. Huh? It's one cell. It has an eye with a cornea, a lens, a retina, So it's one cell that has an eye, no brain. We don't know how it processes information. And then it has this little thing that we have no idea what it's used for. And also, it has these ballastic nematocysts, which are little organelles to kill prey. But we can see that it's following some prey, and it can shoot at the prey very efficiently. And this organelle is more complex than any of the one you can find in Hydra. So this cell, I don't know any. I think this is the most complex cell I know, and you don't have any, comparison in animal. The animal cell types are super simple compared to any of these cells. That's why those cells are super big genomes. They are super complex. We have no idea about their function in the ecosystem for most of them. This is a big, big challenge. In Tara, we also have seen, through the barcoding and using this underwater video profiler, that these cells called Collodarian, they are super big things. They actually, I think a lot of the plastic is probably this that you see on your picture, because when you jump into the deep blue water in the middle of the Pacific, you have tons of these things. And they can be more diverse and abundant than animals in all the tropical and subtropical ocean. So this is typically the thing we had totally overlooked before, because we are not looking at larger size fractions. So protists and eukaryotes are a big, big deal. We are very embarrassed, and it's even worse in forest. We are exploring forests, and it seems that's where the diversity of life is, and frankly, we are very, very few people studying them. So now what we are trying to do now is to use graph methods and method inspired from systems biology to put all this data together, because we have images, genes, ecological data. So we are trying to use, the complex methods to build up interactome of all these elements together, and to analyze their shape, their structure, to the modules that are reacting to some parameters of the ocean. And so we are really thinking a lot about these, and many people in ecology are doing this. And I think we have a good data set for this. So people from artificial intelligence now also are very interested in the Tara Ocean data set, to compare also it to satellite data, just because it's a homogeneous data set of several layers of heterogeneous data. So the data set is good per se. It's quite clean. And so what we have seen is that biotic interaction seems to drive the systems more than abiotic influences. So basically the relationship between the organisms seems to be the key to understand this ecosystem. Like I showed you before on the shaping of a cell or an organism, that's the cohesion, the collective behavior between these species that is probably going to explain the resilience and the shape of this ecosystem. And then we try to, yes, like I said, to divide this interactome into modules and to compare which module of these interactomes are reacting to parameters to abiotic parameters. For instance, we saw that this module is the one that is really correlated to the carbon pump, which is super interesting because it's not one species. Often people were trying to correlate it species by species, which one is correlated to such process. Here it's really a piece of the ecosystem that is correlated to the carbon pump here. And the species that are in this module are often not the one we believed were important for the process. And then this is work by Daniel Richter. I don't know if Daniel is around. But what we are trying to do is and this is really the complexity of the ocean, is that in the ocean, of course, the system is moving. Everything, the ecosystem is in perpetual move. So you need to integrate the fluid, the dynamics, the fluid of the ocean. And this is super complex because we modeled for each Tara Ocean station, we left, in an artificial ocean, drift some particles, and you can see after 100, for instance, if you put particles here, thousands, after 100 years they are there. If you start here, they are there. And so you can model the Lagrangian dynamics between the stations, and here we found something I think quite impressive. We found a sort of law that up to 1 year, the Lagrangian distances explain a lot the erosion of the community. And after 1 year, it's more the environment that explains the difference between communities, the dissimilarity. So that there is some sort of universal law of mixing physics and biology. So I will go toward the end. I think so. What about what for? So we are 10 years later, right, with this data set. It's super complex, and now we are structured in France. They allowed us to make a fédération de recherche, and we are trying. So it's complicated already to assemble all the tutelle and the institute in France. So now we are associated with EMBL, and we will try to make a structure. I'm not sure if it's going to be this Fédération de Recherche, but we are trying to build a structure for the Tara-Ocean Consortium to continue existing in the future. And really, our dream is to go to model, and this is modeled by Mick Follows. This stochastic model by Mick Follows, where he leaves some plankton auto-organized with mathematical rules, is exactly what Eric did at the cell level, but for the big cell, the planet. So basically, Mick puts the conceptual basis, to model seriously the global ecosystem. But Mick is desperate because he has no data. And in fact, he comes sometimes to the meeting and he sees that the data are not increasing that much. So that's, I think, the long-term dream. I think we need another 10, 20, 15 years to reach this, to have enough data to start serious modeling of the plankton. And the plankton you have to consider is that as a whole. That's the problem. Huh? You cannot really, for me, things that are essential to develop and that we will try to develop now, one is what I call ocean phenomics, is we need to develop, and this is really what EMBL can bring in the game, and we'll have a lot of people from EMBL in the next days, Rainer Peppercock, Yannick Schwab, and people that are incredibly good in imaging. And now we really want to work together with them after this omics stage of the project, 10 years, which of course we will continue, but we need, I think, to describe really the 3D dimension of these organisms and to link it as well to, so we have a method to automatically scan in 3D the plankton. It's called EHCFM. We published it, but we have not used it a lot. And so you can see the cells in 3D, you can see the symbionts, you can quantify the organelles. So you can really have some functions, some biogeochemistry of these cells. And then, of course, so a lot of symbiosis. So they make these beautiful things that we are exhibiting in the Planktonarium, and that I invite you to go. And so we need then to link the observation of these eukaryotic cells, complex cells, which have, as you have seen, are making much of the diversity, to single-cell omics, single-cell transcriptomics, single-cell, and then it will come back then to the metaomics game. And so we have a project to do that now. And I think beyond, thinking about technologies we need to do to better understand this complex ecosystem, I think the big need is sampling, because Tara has been superb, but it's a few hundred sites, So we need to sample this planet correctly. And I think very few people understand that, in fact, the big problem is that, for cell biology, and the 50 last years, people were studying their Drosophila or their bugs because they had their system in the lab, right? And when you study the whole planet, you have to homogenize the measure. And this is a big problem. I mean, the only thing we did really good in Tara and in Malaspina, I guess, and in other big expedition, is that we sampled strictly the same way. Whatever the way, the way is not that important. We just did the same way. Of course, if many people can think the same way, it's even better. And so one project I'm really trying to push, and I think it starts to be a bit listened to, is the sailors and the mariners, in fact, not only the sailors, even the people working on big cargo ship. So this guy is a crazy French person, and he sailed the world with his chicken. Now he became super famous. He's on all TV show in France. His name is Guirec, and he left. He was 20 years old. He wanted to leave his family to explore the world. And So he's doing these incredibly nice movies, and he's totally, I mean, he almost died 10 times. He was by himself on the ice, and his boat was cracking, and I mean, it was, Tara was really extremely safe compared to this. And so Guirec, but there are a lot of people like that. I mean, not, he's an extreme. But in Brittany, I can tell you, I mean, we know plenty of people who are sailing the world. And these people are often getting bored on their ship, so we absolutely need to work with them. Because I think, so what we are thinking of now is to develop super simple kits. And I will show you some development because I think it's really interesting. To equip either sailing boat or cargo boat. You know, the network of cargo boat is just absolutely scary. The traffic of cargo boat in the ocean, I don't have the map, but it's the most intense. You know, it's what globalization is, cargo boating, right? I think the 20 biggest cargo boats, I was told, pollute as much as all the industry of cars and planes. The 20 first big thing. So what is really, you know, fucking up this world is cargo boating, because that's where you transport also all the matter and and so the idea is to develop a super simple kit, and we have proved that it works, and really our dream here also that we want to bring is to miniaturize even more, you know, what we did in Tara Oceans, and to and I think what is super important, as you said, Mr. Director, is to you know break the boundaries between science and scientific and societies and makers and the society. And frankly, also, I think Tara worked also because we broke this boundary between the sailors. You know, Eric Kersanti was cleaning the toilet on board the ship, and we had this very particular relationship with sailors. There were many weddings, many kids born from this accident from Tara, mine, and I got 3. And so you know, it was a real human experience. So we broke up this stupid, okay, we are scientists, experts in our lab, and we never see anyone. So, and I think this is so important, right? And so the dream, I think, of this Plankton Planet project is to work with the mariners, because the mariners are the people who really see the planet. You know, all these people at sea, they are very different. They are really obsessed by the sea, and they see they always see that they are like in the space. You know, they see really so they are super sensitive to the planet. So they have a voice. So we need to give them the voice. I think that there are people who can really tell us something. Then we have the scientists, us, we're crazy, obsessed. and then there is a super interesting community, which are the makers. So Thibaut, are you here? Thibaut. Thibaut is a maker I met 2 years ago, and he will be around for the 5 next days. Thibaut is a genius, and you will see what he has done in 3 months. I finished the talk with Thibaut's work. Basically, Thibaut opened my eyes that we need to work with these people. They are super inventive, super nice. They have time, they can work, and so I think by breaking the boundaries between these communities, we can really develop what we need. So what we need is to sample at high speed, because the boats, they don't want to stop, usually. So now we are developing, on Tara Pacific, we did a super tight sampling of plankton at high speed, and it worked beautifully. Thanks to Gabi Gorski, who is so Gabi built up a high-speed net. So we have a protocol, it's published very soon. So it was a high-speed plankton sampling for the whole expedition. So we have a super tight, the data is beautiful, and then we are developing now with Fabien and other people a high-speed net for small boats. It's in process. Once we can collect plankton at high speed, we need Thibaut. And Thibaut, supported by people. This is Manu Prakash. So he's a professor in Stanford. He invented this Foldscope. It's an origami microscope that costs $2. He's an inventor that just go on Google and type Manu Prakash. He's just amazing. He's a star in Stanford, and he invents very cheap things to help a maximum of people. And Thibaut disappeared. He's very free, Thibaut, so he disappeared from my team. I thought I lost him. In fact, he came back 3 months later with this object, which we call now the plankton scope. We are going to publish it also very soon, this month probably. And it's different layers of instruments in computer. Lens. Another lens. This is something to focus, and you pilot the instrument from your cell phone. This is some light with different lightening process, and this is a peristaltic tub. So you can either use this plankton scope to have fun, to make a really nice image of plankton or movies or whatever. And people on boats, they love this, you know. So they will really see things that we never see because they will be obsessed by plankton, like we are. And then you can use it as a flow cam. And so I will show you the quality of the image. So this is a culture of Noctiluca running through the instrument. And at the end you have, so this is one culture, and at the end you have this sort of quality of image, which is much better than the flow cam. So the instrument is better than flow cam. It costs less than $200. Okay? And so the idea is to make a modular ecoscope. We call it the plankton scope, but the idea is to open the plan to everyone, and then to try to force people, not force, but inspire people to develop, you know, a module that can measure, I don't know, plastics, chemistry, carbon, or whatever. So we want to have really a little design to measure aquatic ecosystems for everyone. And I think the magic of this is that if we keep the system, you know, running really homogeneously, because this is about homogeneity, you can use also, you can work with all the makers or whoever is inspired by such a little ecoscope. All right, so I'm finished, and I just wanted to say that it takes a village to go global, and to thank, but it's super to be 10 years later, I think, in Barcelona. It's kind of crazy, and we are still running. So yeah, thank you very much. So I don't know if you are brave enough to discuss a little bit.
So I don't know if anyone want to comment something or tell anything to the speaker. So we will pass some microphones. So please feel free.
Marta.
If you want news about the last result, I can give them to you.
Thank
or
you
to
very
André as well. Huh?
Thank you. No, I don't know if this is. Yeah. Thank you very much. I love it. I love the project and your talk. It's good. You have a very fast excursion for all the findings that you achieve with these expeditions. But I'm in particular curious for one of the graphs you put, and you probably you passed very fast, about how the diversity of eukaryotes in comparison with prokaryotes was so much higher.
Yeah.
It's something that I didn't expect.
Yeah.
And
I think for me, it's the most, but I'm a bit biased, I should say. But for me, it's one of the most important results that can really change the paradigm of ecology. Is that, and actually, so Matt was shocked by the fact that viruses, genes stop at 1 million protein clusters. That's it. And now we have sequenced, many more, and it goes a bit higher, but it's not much bigger. Then Shinny looked at the same, the metagenomes of bacteria, and it goes to 40 million, and now I think it's at 50 million. But basically, we sequence all the mesopelagic realm, the polar, now circle. So I think we are close to the end. So it's 50 million prokaryotic genes. And in eukaryotes, we are climbing. So it's 200, 300. It's logical. So what our belief is that the same cause that, you know, these biotic interactions in plankton. Eukaryotes, they are the first organism that can eat. So when you eat what you are, right? You are what you eat, sorry. So we all know that, and so basically they eat, and they, I mean, we are in symbiosis when our chloroplast, our mitochondria are bacteria, the chloroplast of phytoplankton are cyanobacteria, and they continue this process. You know, now, yes, one thing we have discovered lately is that most 30% of the cyanobacteria in the plankton, no, never mind. 30% of the cyanobacteria in the plankton belong only to the larger size fractions. And we have done a lot of single cell picking, and those are all sorts of symbiosis from ectosymbiosis to medium, you know, ecto-endo to endosymbiosis. So this is a running process, you know. This never happened just once in evolution, like we read in textbook. You have, so I think, and this we had a lot of discussion with Eric, evolution, the evolution of biodiversity, as we know it, start with symbiosis. And then you need to understand what are the mechanisms that fix these superorganisms, or that are maybe not superorganisms yet, but that works very well in the physics of the system, into the genome, and not the other way around. And neo-Darwinism was always the other way around, right? You think about little mutation, making little changes, and then selection, killing the bad guys and keeping the... This is wrong. This is wrong. I mean, of course, selection is there to select, you know, what is super bad, but the creation of complex life occur by meeting other guys and by love stories, basically, which is kind of cool, I think. And I think that's how you can explain the complexification. Evolution is not. I think it's a complexification. There is no word for that in English. You know, the creating complex life. And it's a ratchet of more and more complex life. And this is evolution. This is what we see. Huh? I mean, before, you know, in 3 billion years, before eukaryotes, there was just the world was just a tiny bacteria sucking nutrients and of course making all this game, but extracellularly, right? Do I answer your question? Or? Yeah. Yes.
Yeah, thanks. It's fascinating, and it's amazing just how much data, of course, you collected, and it's really well. But we started this afternoon referring to climate change, of course, and to the changing oceans. So, are you hopeful that you can ever, in this kind of data set and in this kind of analysis, see change? And how long, or are you seeing it already? And if not, how long would you need to keep on measuring to start seeing actually the change in the ecosystems?
Yeah, no, that's a super good question, because in Tara Ocean we just did a baseline. We did a reference data set. So now it's becoming a bit the reference for a baseline, and it was really the goal of the project, is to build this baseline. That's why, you know, I think now in the consortium we should really not, you know, say, okay, we are done and we analyze the data. There is tons of analysis to do still. I think we should really go back to the ocean and measure. And we should really think about, and measuring biology is super complex. I mean, we are not integrating any biology almost in the model of the Earth system. Nothing. All this GX thing and no biology. Well, this is crazy when you think about it. Huh? There is not a single atom in this planet that didn't go through life at some point. And this planet is fully alive, the resilience of these ecosystems is life, right? That's what we need to, I think for me the next 50 years, that's really the challenge, is to understand the ecosystem level of organization. For me, that's evident. But we are lucky to be just at the beginning of this story, I think, now. Because we have the instruments to measure this, to reach this complexity. For a long time, we had no instruments to reach the complexity. Thinking about it, I think ecosystems are the most complex object in the universe, no? Between atoms and galaxy, I'm not sure if there is anything more complex than an ecosystem. As a level of organization of matter, you know? Because it integrates so many scales, you know, from evolution of genomes to...
So, but the complexity, that also means that it will be hard to measure change, to measure if the complexity is changing, or
I think what I learned from Eric Carsanti, because when they were starting the cell, the cell shaping, they were totally lost. They were measuring plenty of enzymes, and Eric was always telling me, because I was telling him the same, but this is too complex. We never understand anything. And Eric was telling me, no, you will see at some point you measure parameters that if you measure sufficiently the parameter space, at some point you will see emerging some issue. Yeah. And sometimes you can, yes, understand and model very complex things with, but we are still, right. Pep, you don't need a microphone.
No, I don't need a microphone. That's one yet because they are recording. They are recording it. Oh. So, well, cool, of course, and there's always this thing, this thought that comes to my mind that most of this work is like not looking at the processes, at the flows of carbon, nutrients, and how much of one type of microbes are being eaten by other types of microbes. So how do you think do you think models will need that, or models will live without, or they will be wrong just because they don't have that? What's your feeling about that?
I'm really not a modeler, and I hate math. So, no, I don't. I love math, but I am just not good at it. So I'm scared by math. I love it, but my feeling. No, I think we at some point. I think maybe you can start by describing the structure, the dynamical properties of this network. How strong they are. If you remove, you can shake these networks and see. You can attack these networks with different abiotic parameters. There are plenty of things you can do with this structure first. But at some point, of course, I think you need, then when you know maybe what you are looking for, maybe you really need, yes, some rate studies, yes. Yeah, absolutely. No, again, speaking to Eric, because I think Eric had this a bit in his life. He was lucky to really go from the beginning to the end of the field of cell biology, And he was always telling us, okay, this is 40, 50 years. Huh? But there are many more, I think. But at some points, we'll have probably measurements on the float or, on things like that. But we are far from this because it's super hard to collect zooplankton. So I think, yes, we are far, far from automatizing the measure of complex systems. In fact, ecology was done by microecologists looking at big things, and then there was the discovery of the microbiome. Now people are super excited about the gut microbiome, but the gut microbiome is like the ocean pre-oxygen, right? It's a super simple system. It's super nice because I think they advance very well the technology, but yeah, it's another, understanding these complex systems, these oxygenic systems is. I think we are really the first who came into, who tried to design a protocol for this. And Eric was really conscious from the beginning about this, He brought us slowly to this understanding, but yeah. Now we are, I'm not sure if we will have the means to do it, At least the Tara team, I have no idea. Maybe we'll just be, témoins, getting the... we are trying, but now we are totally, we are like ground zero, no more money. That's why we are here, in fact. We are basically, we are starting again. So the group is still alive. We have a great pleasure to see each other, but we need to fight funding, and it's not, it's hard. Okay, well, thank you very much. Yeah.